Okay, I'm recording.
Okay, thank you. And I will just, whoops. I want to go see if I can get my pointer to be a laser pointer. Okay, so I'm really excited about this. It's great fun. It was actually a torment to decide what to talk about with you guys, but I decided that I had two missions. Mission one was to give you, those of you who are relatively new to the SDG, a bit of an overview, historical overview. And I think those of you who are old with gray hair, etc., who were there at the beginning, should chime in whenever you want. Astrid, what are you scratching your head for? You have gray hair?
I'm looking at my gray hair.
Should chime in whenever you want. Please feel free to interrupt because this is our SDG family lab meeting, right? So I wanted to go back, since we're recording it, to the very beginning for those of you who don't know where we came from. And I'll spend a little bit of time on that. And that part of this probably should be required watching or thinking about for any new people who start in our labs, because it can set some intellectual stage. And then I didn't know what to do after that. So I had the brilliant idea of just picking 10, I thought about what are 10 or 12 or 15 of my most favorite slides. So I went and I found some of those slides in various and sundry talks, and a few others I couldn't find, and I didn't really make them. So that's how I came to decide what to mention, which it was sort of random in some ways. It's what I came across in searching through slides. And as I said, feel free to interrupt. Now, this is our SDG lab meeting, which I hope is going to continue. So I thought I'd introduce my present lab. My present lab, they're postdocs, Leandro, Srinivas, Sono, Katya, Jason, and Yulian. And half of them are theorists and half of them are experimentalists or something like that. And then I have two PhD students left, two recently graduated, Dan Powell, who's now with Patsy, and Dave Hampton. And now Mara and Hillary are still with me, with us. Two fabulous research staff, Peter and Caitlin, and some of you have heard from Caitlin, I'm sure. And right now, we have 4 undergraduates who are working this coming year. We used to have many more, but COVID's taken care of some of them. They're not sick, they just aren't allowed to come back yet. And I'd just like to tell those of you who don't know that I really don't want to call, they're not really mentors, but I worked with Al Selverston for my PhD, David Barker as a postdoc for 1 year at the University of Oregon, and Jacques Souquillo in Paris as a postdoc. Okay, so and I just wanted to, before going on, and maybe other people will remind me, we remember as part of our community, Pete Getting, who was extremely important to the small circuits, simple circuits community in the beginning, and he now died. And losing him in the late 80s, as we did to a massive stroke, was incredibly difficult for our community because he was really the de facto leader. And I think both Rons, you would probably agree with me, and I still think of him a lot. And any of you who've not read his work, in particular his annual reviews paper, published in '89, should do that because he really outlined there a lot of the concepts that we're still all working on. Now, Don Maynard, you'll see in a moment, was really the father of the SDG preparation. And Maurice Moulin, and he died in probably 1972 or 1973, around then. And Maurice Moulin was the leader of the French part of this contingent, and he died, I don't remember exactly when, but a while now. So, and doing this reminded me of them. Now, in the beginning, and this in the 1960s, people were looking for preparations with which to study how the brain works. Sound familiar? In the early days, simple systems were called simple systems. And there were books and meetings called simple systems. Now we all know that none of them were ever simple, but people thought they were going to be simple. But what Don Maynard and that cohort was looking for were systems that were amenable to electrophysiology, where you could identify neurons, find their connections, and in particular, central pattern generators were very attractive because they produced rhythmic motor patterns that were ideal because they had defined outputs. You had a measurable output, so you could tell what the thing was doing, and you could ask the question, if you make an in vitro preparation, if you take it out of the animal and put it in a dish, is it doing something that's recognizable in terms of what the in vivo preparation would be doing? And that was extremely important. So some people started working on sensory systems. I'm not so much in the SDG at the beginning, but the motor side was really because if you worked on a rhythmic motor system, you had an observable output. And in the early days, people worked on crickets and cockroaches and leeches and locusts and crayfish and snails and turtles and tadpoles and etc. A whole variety of preparations. And they were sought because they had features of these general principles. So when Don Maynard, who developed the SDG preparation in the late 60s, early 70s, and by the way, when he died, he was just starting the study of graded transmission of inhibition. But he really wanted a preparation which had a small number of neurons that could be identifiable uniquely, and that where he could record intracellularly and see synaptic potentials and see action potentials. And the SGG neurons were better than most insect systems at the time because the action potentials were big and the synaptic potentials were really big. And so you could actually imagine wiring up the circuit. Now, a little bit of history. He taught Malcolm Dando, remember? Maynard and Dando, the Bible, the anatomy Bible that's floating around. Don Maynard taught David Barker, and you'll hear a little bit more about him, because David went as a faculty member to the University of Oregon where Don Maynard was. He taught Al Selverston in Bermuda the SDG preparation. He taught Dan Hartline in Bermuda the SDG preparation, and he taught Maurice Moulin, and I think Maurice was a postdoc with him or something like that. Patsy, am I right about that? She's muted, so, but she's nodding yes. She's nodding yes. So the preparation really came from Don through these lineages, and all of you ever since has actually come via some pathway through one of these lineages. And we have some very complicated family trees. And just to give you one little amusing joke, Al Selverston, my advisor, was a graduate student with Graham Hoyle, and Patsy's PhD advisor, Dennis Willows, was a graduate student with Graham Hoyle. And therefore, Patsy and I related that way. But then Patsy did a postdoc with Frédéric Nagy and Maurice Moulin. So we are also have these other complicated family lineages. And all the rest of you also have lineages with one or the other, or many of us.
I hope all of this is on NeuroTree.
What?
I hope all of this is on NeuroTree. So,
I think that's not good, whatever you did. Astrid said she hoped it's all on NeuroTree. I don't know what's on NeuroTree because I'm not a very good, but anyway, if someone wants to make sure it's all there, you can ask us. Anyway, so I'd like to go back to UCSD in the early 1970s. So there were two SDG labs at UCSD when I was there, Al Selverson's lab and Dan Hartline's lab. And if I looked and remember all the things that were happening in the Selverson and Hartline lab together, every single problem that people are studying in not every single problem, but a large fraction of the problems that people study today were already presciently being studied in the Selverson Heartline labs. So Al, when he was a postdoc, worked with Don Kennedy, and he worked with Procyan yellow dye cells. And so at the time in the lab, he was filling cells with dye and they were being reconstructed, and Cheryl Glasser was working on those reconstructions. David King was doing EM connectomics at the time. John Miller and David Russell were doing mechanistic circuit analysis. I started the work on transmitter neuromodulator identification. And then Dando and Selverson, the first IVN command fiber paper, was the first of the physiology of modulatory projection neurons. And that preceded, just by a little bit, the work that was happening in France that Patsy and Frédéric Nagy did in APM and other cells. At UCSD in the early 70s, Heartline's lab pioneered biophysics of ion channels and STG neurons. And he was constructing biophysical models. And in particular, he was interested in the role of IA in controlling the latency of PY firing. And he had all sorts of models. I mean, there were two papers published in 1979, which were some of the first serious papers of that sort. So if you think about these themes, many of us are still following up on many of these themes, but they really were already embedded in the Gestalt. And I have to give Al and Dan credit for actually seeing extremely clearly what the essential problems were going to be and what the goals had to be. Now, they didn't have the tools. Dan's computer was filled a room that was bigger than my office here, and it had to be air conditioned and this, that, and it probably, I don't know, probably at 64K. I mean, it was really very, very difficult. So both Dan and Al were always pushing the limitations of technology. And so when I look and see the technologies that we have available to us today and what they were struggling with, it's remarkable how daring they were and how much progress they made, or we all made. Okay, now, the Moulin Lab in Arcachon in the 1970s pioneered sensory inputs to the SDG and the characterization of descending modulatory neurons, and Patsy was part of that. The Barker Lab at the University of Oregon in the 70s pioneered studies of amine modulation in the STG in the heart. David Barker started out in Ed Kravitz's lab working on octopamine, and then when he went to the University of Oregon, he started working on dopamine, octopamine, and serotonin in the STG and other places. And Teddy Maynard, that she was Don Maynard's wife, was doing EM in the 1970s as well, and she continued for a while after Don died. Okay, so those were the beginning themes, and I think, it's pretty interesting if you think about connectomics and field of neuroscience today, what was there. Now, I just want to step back and tell you about the important role of the first and continuing STG meetings. The first STG meeting was held in 1984 in La Jolla, and it brought together the whole community for the first time. It was organized by Al Selverson and Maurice Moulin. Now, I should tell you, before this time, there was a great deal of, I wouldn't call it ill will, but let's say collective suspicion. There was the French lab, there was Sasaki was working in Japan. There were random people working around the world. And since we never saw each other, it was very easy to demonize the other group. Now, we were all working on different species, and that's probably important because people were seeing different parts of the elephant, if you will. But that first STG meeting made it very, very clear how important it was to get everybody into the same room. And that's why I think we should continue to be in the same room, if not virtually. That first meeting led to what I call the Green Book, the Crustacean Stomatogastric System that many of you know was published by Springer Verlag, and it was prohibitively expensive. It was followed soon after by the Purple Book in 1992. notice I call them by their, which was published by MIT Press, which is probably still vaguely, if not in print, at least available. And then somewhere in the middle or late 80s, I don't remember what it was, I wrote the first of several HFSP grants for the whole community. And as part of those HFSP grants, we initiated yearly STG meetings. And it was those meetings that started pulling the community together. Now, after we had the HFSP grant for a while, then Scott Hooper wrote and obtained an NSF grant for the meeting that continued, and then somebody else, I think, did it even after that. So when people ask me, how is it that the STG has managed to maintain itself as a preparation and as a community over the years when so many of the people working on the other animals were lost, I think it's precisely because of these STG meetings and the notion that we had an international community that really helped maintain it. And that's why I'm so enthusiastic now that we can do this on Zoom to think about really always getting together as a community, because as I said, it's really hard to dislike the people you know. It's much easier to dislike people you don't know. Okay, so the challenge for those entering the STG, I estimate, and I haven't counted, but I just looked at my EndNote library, and maybe Farzan has this number for real, that there are about 700 to 800 papers in the literature using the STG. Many of them are elegant, long, and very detailed. Now, it's hard to ask a new student to read 700 or 800 papers. And that's why all the younger people in the field don't know anything. Now, they all know a tremendous amount. But I'm always frustrated because I know there are papers that they haven't read. And then when I looked at this and I said, well, how could they? Right? So I think one of the things we should maybe talk about as a goal of these meetings is how to somehow or other manage to get beyond the difficulties of this large literature, which is, however, very important for us. And my hope would be that the joint SDG meetings will help create paths through that literature. Okay.
In defense of the younger people, I remember one or two instances where you didn't remember your own papers.
Well, I'm sure. I'm not criticizing them. I'm saying they have an impossible challenge. Yeah. How about you? You remember all your papers? No. Okay. So
I also want to say, if you just read any review article that Eve has ever written, then you're all set. I mean,
not so much. Not so much. But
Mikey remembers everything, so if we just hold on to him.
yeah.
I remember having a stack of papers about that thick of Maynard and Dando, and I read through them like a book, and I was just like reading every second of the day, just like, oh my God, I'm never going to finish this on my first day at Jorge's lab.
And you didn't. Because I mean, I think that some of us who've been around for 40 something years probably have read most of them and then forgotten many of them. So the problem is I remember better the early ones than I do the ones that were published 5 years ago, not so good. Okay. So the partial list of topics, and I think we should think about these as sort of organizing principles. Circuit cracking and dynamics, pyloric and gastric mill rhythms. And that started in the very beginning. Single cell and half center oscillators, amine and peptide neuromodulation, peptide identification and localization, characterization of modulatory neurons, biophysics of conductances, rated transmission, function of sensory neurons, dynamic clamp and what you can do with it, computational models of neurons and circuits, development, molecular characterization, single cell, etc., homeostasis and neuromuscular function, in vivo recordings and alter environments, oxygen, pH, temperature, etc. And I'm sure there are other topics that should be on this list but aren't. But if you think about it, you can see, again, we're spanning a large portion of sort of modern neuroscience, but in a particular context. Okay, now I just want to start with some of my favorite slides, but obviously not all. There were some I couldn't find. And apologies to all of you whose wonderful work isn't going to be specifically highlighted, but then we have next month and the month after and the month after and the month after. Okay, favorite slide number 1. That doesn't mean it's my most favorite slide. It just happens to be, this is a random order. It's not quite random, but it's pretty much a random order. But this is my favorite slide number 1, which is Lin Jun very graciously updated this slide just yesterday for us. And I love this slide. And I'm going to tell you why I love this slide. I love this slide not only because it makes a very, very important scientific point, which is that the STG is multiply modulated, that many of the same substances are both hormones and found in the inputs to the STG, etc. But I appreciate this slide so much because this is a not totally complete, and probably will never be totally complete, but a very close to important answer to the question that I started when I started my thesis. What I wanted to do for my PhD thesis was to work out all the transmitters and modulators in a functional circuit, because at the time, people were studying serotonin over here and dopamine over there and GABA somewhere else and acetylcholine somewhere else. And there were whole meetings about serotonin and whole meetings about dopamine and whole meetings about GABA, but nobody was trying to figure out what all of these multiple transmitters were doing in the function of a circuit. And I said to myself as a second-year graduate student, well, I don't understand why you need to have so many transmitters. And my advisor said, well, you just need excitation inhibition. What do you need all those transmitters for? And so I said, well, there must be some reason. And so I thought that the only way to figure it out would be to try and characterize all the chemical signaling molecules in a complete circuit and then see what we learn by seeing the whole. And that's what this shows you. And the answer that there are many, not one or two, and that they're complicated and that they're interesting, and that we have here fast signaling neuromodulation and slow neuromodulation, and then multiple versions of all these peptides. So obviously this took years and years and years and years and years and years and years of work. And the other reason I love this slide is because many of the papers that went into this work are sort of boring papers, but this is not a boring image. And so this is an example to me about how sometimes you can get a really important big picture answer by doing a lot of boring descriptive work.
There is a problem with this figure though, Eve.
Yes.
You ran out of colors.
No, I didn't make the figure. June made it.
Well, and also I have to say that Kellen helped to update it, over. So you see the numbers of these isoforms getting more and more. Yeah, so clearly the color, if we want to make them all color, then that would be a problem. Yeah.
A over here, 36, that means that 36 different chemical isoforms of that.
And for murfamide is 41 now with Kellen's new work on some of these. So.
Right here. And but that's in the pericardial organ probably, or the eye stalk, whereas what's been found in the STG so far is a little less, but that's probably because it's been less intensively studied. Okay, so as we move along, now we know all about transmitters and modulators and all of them. Just a few more favorite slides. This is one of my early favorite slides. This was Judith Eisen's thesis. And what she did, among other things, look at the effects of dopamine before Ron's lab on the ABPD neurons. And she was able to show that dopamine appeared to excite the AB neuron and inhibit the PD neurons. And this was the first instance that I know of where we saw that the same modulator could do opposing things, functionally opposing things to electric coupled cells. And you see that here. These are control recordings, PD, AB, PY. And in the presence of dopamine, you can see the slow wave is decreased in amplitude. Here it's increased in amplitude. And here the combined IPSP is shorter. And that's because the cholinergic part of that joint IPSP seems to be truncated. And in the context of this, she argued that this was a possible contributor to changes in firing phase of the PY. But dopamine, I'll just show you one more. One other thing that I loved from this general era was that Judith was first able to show that a number of different modulators could turn on an isolated AB, which these data were from Pennularis interruptus, which when completely isolated, the PD cells were killed and the front end was removed. The AB would often go silent. And when she applied pilocarpine, which is a muscarinic-cholinergic agonist, or serotonin, or other things, you can see the oscillation is turned on, it gets faster and faster and faster. Now at this point, Judith, this was all done on a chart recorder. Judith pushed the button on the chart recorder, and now you can see these beautiful large AB oscillations at a faster speed. So this is what we'll call modulation of intrinsic properties, and this is just, in my mind, one of the early pretty examples of that. Now, following in 1985, Ron, was it 1985 when you joined us?
I
Something like,
don't know. Yeah, 85 maybe.
something like that?
Yeah, 84, 85.
Yeah. Yeah, well, wait, maybe a little earlier, because
I, ended up in Cambridge in 82.
Wrong Ron.
yeah, wrong Ron. I was talking
Oh.
to Mark Ron.
Oh, sorry.
Yeah, I think it was 82 or 83.
Yeah,
I basically I saw you gave a wonderful talk at neuroscience and talked with you about switching away from what I had been doing to STG, and you very, very kindly invited me down to your lab and taught me and Bob Flam how to do the prep, and we did our first collaborative work on serotonin there.
and that was
in the early 80s, 82, 83, I think.
Yeah, it would have to be, because the serotonin paper with Barb was 84. So it would have had to have been then. So at the time, Ron had been working on amines originally out of Ed Kravitz's lab. And so he kept going with the amines, so serotonin, octopamine, and dopamine. And we started focusing on the peptides. And that's how we sort of kept out of each other's way as much as we could. So this is one of my favorite slides. This is a somewhat redraw of another very nice figure that Ron has published that I find very hard to parse. But here what he did, his lab did some very beautiful and careful work looking at the effects of dopamine on receptors in each of the pyloric network neurons, and made the very remarkable finding that every neuron had dopamine receptors, but those dopamine receptors did different things to the voltage-dependent currents in the cell. So I think this is one of the most important findings in neuromodulation that was made in the STG. And I think it's one that all of our vertebrate friends should know better than they do know. And I think, Ron, don't you agree? Yes, you must. And so, and this other slide is just a partial list of substances that act on the LP. And so the two messages here is that dopamine has very interesting convergent and divergent actions on specific identified STG neurons, and here that a single neuron can have receptors for a very large number of different substances. So, and these I think fall into the category of general principles. And this is another one of my favorite slides, because this is the Pennularis wiring diagram. And I think Ron's work and the rest of some of our work and Ron's work, in particular Ron's work, has shown that each neuron has multiple currents subject to modulation by many substances. and each synapse is subject to modulation by one or more substances. So again, I think this is a very important thing. Now, when Ron and we were doing this work, the vertebrate world was still thinking of, I don't know what they were thinking, but they certainly were thinking that neurons would only have receptors to very few substances, and that whole behaviors would be triggered by single modulators. They still think that. Now, in the category of favorite slides, when Jim Wyman made this slide, and this was in Crab, where everything else I showed you so far I think was in Panularis. What he was trying to do was show in one preparation, these are recordings all from the same animal, and he removed the front end, he washed it very extensively, and he found one of those preparations that went silent or virtually silent in the absence of the front end. And then he sequentially applied pilocarpine, that's the muscarinic agonist. He washed it back to silence, serotonin, he washed it back, proctolin, dopamine, SPRN, TNRN, flumerfamids, CCAP, RPCH, and then silence. I don't know who's binging at me. Hope it's not. Anyway, when we first got these data, we focused on the take-home message that different modulators could produce different forms or different patterns from the circuit. So we thought about it as mechanisms of plasticity. And then over the years, other people and we realized that not only do you have plasticity here and different forms of the pyloric rhythm, but you have tremendous, if we want to call it degeneracy, and that is many different substances are able to turn an off rhythm into an on rhythm. And that although they're all a little bit different, they're not as different as any of them, except for dopamine, come back to that, is to the starting condition. So I think we can in 2020, I like to think that yes, these modulators can shape the rhythm and produce different patterns, but they also provide mechanisms for maintaining stability. That is to say, the animal really needs there to be a pyloric rhythm, and it's much more important that it have one rather than the actual details of how it's working. Now, in these recordings, Jim put on a high concentration of dopamine, and you'll see that these recordings are what I would today call a crash. That is to say, you lose the alternating pattern of activity between LP and PD. And this is a more extreme perturbation, and obviously CCAP and serotonin are giving you partial sort of debilitated rhythms. But dopamine is, a number of you have studied, does all sorts of things to spike initiating zones in the PD neurons, etc. And that's why at high concentrations you see this. Now, the deep question is, of course, we go back to this, how if every synapse and every cell is subject to modulation, why you actually get a stable response as these are, and why you don't get crashes all the time. Because as we now know, you're starting out with different balances of conductances, so it's actually remarkable that any of these things are as stable as they are. Okay, continuing on with some of my favorite slides. This comes from Don Blitz and Mikey, and where they were studying all these beautiful descending inputs, and this just makes the point that you have 3 different identified descending neurons. Each of them has proctolin, 2 of them have GABA, but when you look at the patterns that they evoke, and here you just see it in cartoons, each of them is different. So not all proctolin containing neurons evoke the same motor pattern, but there's obviously a real difference in the way they do so. And Mikey knows a lot about this, as does Dawn, more than I do. Okay, now, again, in not necessarily any real order, this is a segue into the problem that I think has been extremely influential in the field, and that this project started, and I blame Jorge for this. Jorge, you there? Yeah.
Yes, I am. I was muted.
I blame Jorge for this. And
Why?
that is why. So when Jorge was doing his thesis, he set out to use voltage clamp to characterize the voltage and time dependent currents in a neuron, and he chose the LP neuron because there was only one of them and it was big. And he did a beautiful piece of work characterizing the currents, and then he and a postdoc in Herb Epstein's labs started to build a model because they wanted to build a model that would be a good LP model. And Jorge had done this really elegant biophysics. And the model became the curse of 1991 to me. And I'll tell you why. And it became a curse of 1991. Now, Frank Buchholz, who built this model with Jorge, was writing in Fortran and running on a mainframe. So it wasn't so easy to tune this model. This is 1990, 1991, you can think about it. And, you know, he had to turn the code in and then come back the next day and see if it had run. So tuning it was slow. But after 9 months, what we had learned from that work was that the two things that we really couldn't measure at all, or at all well, were the most important for the way the model worked. And that is, number one, the capacitance, and number two, the sodium current. Now, you all know, you all remember that the sodium channels are way down in the axon, and they're not really voltage clampable from the cell body. So we didn't have, Jorge was unable to measure the sodium channels from his electrode voltage clamp in the cell body. And then the two things that seemed to be most influential in how this model worked were the two things we actually had to make up. Now, you can have a long conversation with Jorge offline or another lab meeting about the difficulties of the right way or the wrong ways of measuring capacitance. But be that as it may, I found it very disconcerting to know that I couldn't believe those numbers necessarily. So eventually, Frank and Jorge got this model to more or less work. And by the way, I should say, when we published it, people said it was beautiful because it was one of the best conductance-based models in the literature at the time. And everybody I talked to at the time said, oh yeah, you know, these models are very fragile, they're very non-robust, and we were at least using data from crustaceans, but people were building models using data from squid and guinea pig and eel and this and mouse and putting it all together at different temperatures, etc. So we were far more disciplined than most, but still it was very unsatisfying. So it was very unsatisfying to me because I kept on saying, you know, the LP cell gets it right all the time. And if I've got these really smart graduates and they're really smart postdoc and it takes them 9 months to tune this model, why is the LP cell getting it right? Anyway, I nagged Larry Abbott for a year about this problem, literally. I have lunch with him almost every day and I nag him every day. And he got really pissed at me one time and he said, stop it, it's too hard, this problem's too hard. And then I kept nagging him, and then eventually he came back with a very simple idea. And that is that there could be a negative feedback homeostatic type system, which is what biology does all the time. And the real question that I was nagging him about was, how does the LP cell, which lasts in our crabs for, I don't know, 8 or 10 or 15 years, maintain its function when all the ion channels are being turned over? And ion channels don't live in the membrane for very long, and the LP cell always got it right. So in that first model, which was published by Bernard Lemesson. There was a very simple monitor of activity, which was intracellular calcium concentrations. And it was being used to up and down modify the maximal conductance for the sodium current, for the calcium current here and the calcium activated K currents. And so in this one picture, if this is now a plot over here, if you have a plot of the calcium activated K and the calcium current, his starting condition was here and the model was a burster. And then he perturbed it with depolarization and it lost its activity. It's no longer a burster. Now it's firing tonically. Slowly over time, because that's the way this model works, slowly over time, when the model leaves its target level of activity, it changes the balance of conductances. And now in response to continuous depolarization, it rebuilt itself back into a bursting mode. Now, this model was the first of 3 very different models that my lab has produced, but that's not why it's important. It's important because it caused all sorts of people to think about this problem. We did a little bit more then, and then Gina, when she started her own lab, took this problem of homeostatic regulation into homeostatic regulation of synaptic strength, which she called synaptic scaling. And today there is a literature that's much bigger than the STG literature on homeostatic regulation of synaptic scaling and intrinsic excitability in every animal known. And you know, the lineage was very clear. Gina was a postdoc in the lab when we did this, and she was very good friends with Wendell's. And then when she started her lab, she and Sasha Nelson immediately realized that a very similar way of thinking about things could be used to think about the regulation of synaptic strength. And so most of the people who work on homeostatic regulation of synaptic strength have no idea that this is where it actually started. But I think it's sort of nice for you guys to know. Anyway. One of my other favorite, I'm gonna stop soon. One of my other favorite slides, this comes from a paper that Jorge and Mark Goldman published, is what we call failure of averaging. And so what Mark Goldman was doing is he created at the time of this work, 5,000 model bursting neurons, or model neurons, with, I think there were 5 or 6 conductances in each. And he then simulated all of them and then selected them on the basis of what their activity profiles were. This was a precursor to what Astra did just a few years later. So here you have a single spike burster. That's one. Here's another single spike burster. Here's another single spike burster. But if you now look at the sodium current, it's big here, and the delayed rectifier is small here. In this model, which looks very similar, the sodium current is little, as is the delayed rectifier. And here, the sodium current is little and the delayed rectifier is big. And that's because there are differences in other currents as well. But the interesting thing is, if you now calculate the means of the sodium currents, you get this, and the means of the delayed rectifiers for 150 of these guys, you get this. But if you now start building models from the mean currents, you don't get a single spike burster, but here you get a 3 spike burster. So I love this because it shows you that if you just calculate means and you start building models from mean data, you can actually get something which is different from all of the individuals whose data went into the mean. Now, and that's just shown in the rest of the slide. The single spike bursters are over here in these blue dots, but these other models, model 4 and model 5, are sitting over here outside of that area. Now, I just have to tell you one more funny story about this. And this goes to all the people who will publish means and standard errors of the mean, and then try and model those means. And I think that's a really dangerous thing to do in general. And what we need to do is look at the distributions and the correlations and think about that. But that's a whole other big, long story. I was giving a talk at the cognitive neuroscience meeting, 10 years ago. And I showed this slide, and because that community averages all the time. And so at dinner, I was sitting next to an fMRI guy, a famous fMRI guy, and he said to me, you know, we published a study with fMRI data from 80 individuals, and we gave them this task, and we showed in the mean data that 2 areas in the brain lit up, area 1 and area 2. And he said, then I went back and I looked at all the data for each individual, and 40 of the individuals lit up area 1, 40 of them lit up area 2, and none of them lit up both areas. I started laughing. So then subsequently, I told that story to someone else, and he looked at me and he said, you know, calculate the mean number of breasts and the mean number of testicles in a population of 100 undergraduates. So be that a warning for you all when you think about looking at your mean data. Means can be very, very useful, but they also can be very misleading. Now, I know some of you were laughing, but I can't hear you laughing, so it's unfair. Now, this, of course, is Astrid's extremely famous figure, which you all have seen. Astrid, now this was published in 2004, was trying to build models of the STG. She ran 20 million model networks, and she selected 4 ones that were good pyloric rhythms. And she matched those data to work from 99 lobsters that Dirk measured. And now you can see that this set of parameters and this set of parameters are totally different. So this is a very beautiful example of what we'll call multiple solutions or degenerate solutions to producing very similar behavior. Now, this paper was very, very important for my lab because I took it very seriously. And I said, if I take this seriously, we have to measure things like conductance densities and synaptic strengths in the biological system. But more importantly, what a lot of those pesky theorists were saying to me, that an animal with this set of this set of conductances and this set of conductances should respond very differently to perturbations. And that's why we started doing all the perturbation work that we started doing. Now, this was the work that Dave Schultz and Jean-Marc Goyard started looking at, again, in the LP cell. And there, Jean-Marc replicated data very similar to what Jorge had done a few years earlier, and discovered this 2 to 6-fold range of conductance densities in isolated cells. And I don't remember what I did next. You know, I'm just going to end and not talk about Lamont Tang's data, but I just want to show you something that Sarah showed in 2012. Sarah Haddad, when she started in the lab, and a number of years ago, she was a new graduate student, and so I asked her to repeat some of Lamont's data because I just wanted to give her something to do to get her started. So she made these recordings, and here's the pyloric rhythm at 11 degrees, at 20 degrees, at 25, at 30, and 36. And she showed me these recordings. She came into my office. She said, it's not crashing at 35 degrees. And I said, you have the thermometers in the right place. Are the thermometers broken? Do this, is the temperature right? And she got really angry. I mean, she said, I've got 2 thermometers in there and I'm measuring it right. And I said, and then I said, go do it again. And then she came back and she said, this ganglion is going at 7 hertz. And I said, oh my God, because when have we ever seen a pyloric rhythm at 7 hertz? Well, it turns out that was the the spring or the early summer of 2012, and that was a winter that never was. The ocean was 6 to 7 degrees centigrade warmer than normal, and so what we're seeing here was a long-term adaptation or acclimation of the crabs to this way too warm water. Now, at the time, we never knew whether our crabs had gone to visit Patsy in Maine and we were seeing Long Island crabs, or whether the crabs that were sensitive to temperature had died and we were only seeing the resistant ones, etc. But it was really sort of a little scary to see this kind of recording. And then seasons went by and the crash points went back down like Lamont had seen. And then this summer, we seem to have similar data because again, we had a warm winter and a very warm summer. And so 2020 appears to be heading in the same direction. So I think it's really a very interesting set of questions to be working on wild caught animals. Even though we keep them in the lab for weeks before using them, we see the residual effects of these long-term environmental changes. Now, if you only study them at 11 degrees, you would never know that you have what we call warm crabs. And it was precisely because we were perturbing them that we saw these changes, these long-term changes in their behavior. And I would just like to show you my last favorite slide. This is my favorite slide from 2020. And this comes from a paper that Leandro just published. And this is a model. This is a model burster that we're looking at as a function of temperature. And what Leandro's doing is he's showing you the last spike in a burst. Okay, at 10, 11, 12, all the way up to 25 degrees. And he's doing it using a really lovely way of displaying currents, where we've got the total outward current shown here in black, the total inward current shown down here in black. And now the contribution of each of the outward currents and the inward currents to the total inward and total outward current is shown here. And so in purple, we're looking at the 8 current. You can see it's big here. And as you get warmer and warmer and warmer and warmer, you can see it's getting smaller and smaller. Whereas the calcium activated K, which you see here in orange, starts out really small. And over temperature, it's getting larger and larger and larger. Now, the cell is managing to burst just fine, but the relative contribution of each of the currents to the spike repolarization or the burst repolarization is smoothly changing as you go through this perturbation. And I love this result because to me it's telling you, and we actually saw this same feature in Jorge's 1992 paper. We saw this, that the relative contribution of 2 K currents to spike repolarization would change as a function of depolarization. But what this tells you, it gives you a way of thinking about why we may need so many different potassium currents that have overlapping voltage and time dependencies. And it's so that you can smoothly move between what other people would call different mechanisms and see how that plays itself out. And I think that was the end of my yeah, I'll stop there. As I said, I'd stop at 1:30 or 3:30, whatever it is. And I was going to show you Gabrielle's stuff, but you probably have all seen that too. But I think this is a really important point, and that it may really give us an idea about why so many neurons have so many different voltage-dependent currents that differ a little bit from each other. And it's so that you can extend my hypothesis. So hypothesis, it allows you to extend the dynamic range over which these networks work, neurons and networks work. So that's all I wanted to say. Now, I think you can see that if you, I think I'll stop sharing so I can see everybody's faces back, that a lot of the themes that I mentioned had their beginnings, in the very, very beginning. And a lot of them are still looking at or we're addressing problems that people are studying today in all sorts of animals. And so our challenge is to figure out how to stay relevant. And this is always the challenge of the SDG, is how to stay relevant to the entire field of neuroscience while taking advantage of the wonderful attributes of this nervous system. And so that's how we can help each other figure out really how to really profit from the advantages of the preparation and yet stay relevant. And I think while I do my one last piece of philosophizing, I think one of the really difficult things for experimentalists to do ever is to figure out which of your experiments are studies in idiosyncrasies and which are giving you insight into general principles. And so that's what we have to do for each other, sort of help us stay focused on things that will become general principles and not just be the idiosyncrasies of the crab, because the crab has a lot of idiosyncrasies. And not all of them are in Lin Jun's picture. So I think we should open up to that general conversation you guys wanted to have.
Thank you, Eve. That was wonderful. So Jorge, if you can stop recording it, then we can go into our general